Aging is a huge health, societal, and economical burden that is marked by a decline in an organism's physiological activities. Many diseases, including malignancies, cardiovascular diseases, and neurological diseases, are linked to advancing age. AI has found anti-aging technology. Want to know more? Keep watching! Hi, my name is Alex, and welcome to AI News Daily. In this video, we will be looking at the top AI anti-aging technology. Don't forget to subscribe and turn on the notifications. Let's get started. Though chronological aging cannot be reversed, artificial intelligence may soon assist us in slowing biological aging, which is impacted by lifestyle, behavior, and other factors, and is a more accurate predictor of health and mortality. We can easily guess someone's age by looking at them, hearing them speak, or even smelling them. These are all symptoms of biological aging. Even a layperson's eye, let alone a doctor's, may be able to detect a person's health status if they appear older than their chronological age. AI's ability to absorb and analyze massive volumes of data makes it a valuable ally in the quest to not just discover aging indicators before they manifest biologically, but also to help people live longer, healthier lives. Through patterns in medical data, photographs, and other sources, AI can recognize trends and determine casualties. AI algorithms evaluated daily images of mice to extract probable aging markers and build lifetime management methods in a project with mice. The same visual biomarker learning mechanism can subsequently be applied to other species, including humans. Beyond picture analysis tools, AI can detect biomarkers by identifying recurring patterns in human aging. AI can detect aging trends in different populations by sifting through large amounts of data from various blood tests, retinal scans, muscle analysis, and other sources, or by comparing human data to that of other species. AI can offer health solutions in addition to recognizing prospective difficulties by studying the effects of therapeutic treatments, preventive remedies, different lifestyles, and more. On a deeper level, understanding how proteins and cells respond favorably or adversely to therapies allows AI to aid in the efficient development of medicine. Deep learning's capability could soon assist us in alleviating the discomforts of biological aging. A team of Surrey chemists developed a machine learning model based on data from the Drug Age database to predict whether a compound can extend the life of Caenorhabditis elegans, a translucent worm with a metabolism similar to humans, according to a paper published in Nature Communications Scientific Reports. Due to the worm's short lifespan, the researchers were able to observe the effects of the chemical compounds. The AI identified 3 chemicals that have an 80% likelihood of extending the lifespan of elegans: flavonoids, which are antioxidant pigments found in plants that enhance cardiovascular health, fatty acids like omega-3, and organooxygens, which include carbon-to-oxygen connections and include alcohol. Sofia Capsiani, one of the study's co-authors and a final-year undergraduate student at the University of Surrey, said, "In modern medicine, aging is increasingly being recognized as a series of diseases, and we may use digital technologies like AI to assist slowing down or preventing aging and age-related diseases." Our research reveals AI's groundbreaking ability to aid in the identification of anti-aging chemicals. This research highlights the power and promise of AI, which is a specialization of the University of Surrey, to generate huge advances in human health, stated Dr. Brendan Howland, main author of the study and senior lecturer in computational chemistry at the University of Surrey. Many disorders, such as cancer, cardiovascular disease, and neurological diseases, are linked to aging. An emerging study topic is pharmaceutical interventions that slow down the aging process and delay the onset of age-related illnesses. The goal of this work was to create a machine learning model based on the Drug Age database's data to predict whether a chemical substance would lengthen the life of Caenorhabditis worms. The random forest approach was used to create 5 predictive models with molecular fingerprints and/or molecular descriptors as features. For categorizing the substances in the test set, the best-performing classifier, created using molecular descriptors, achieved an area under the curve score of 0.815. The random forest algorithm's Gini significance measure was used to rank the model's features. Descriptors relating to atom and bond counts, topological and partial charge qualities were among the top 30 attributes. In an external database of 1,738 small molecules, The model was used to predict the class of compounds. The screening database's chemical compounds with a predictive probability of 0.80 for enhancing canorhabditis worm longevity were divided into flavonoids, fatty acids and conjugates, and organo-oxygen compounds. Interventions that target the cellular and molecular processes of aging have the potential to prevent and delay age-related diseases. Using food limitations, genetic alterations, and pharmaceutical interventions, several aging studies have revealed interventions that prolong the lifetime of model species, ranging from nematodes and fruit flies to rodents. In the multicellular species Canorhabditis elegans, Lee et al. Revealed the first evidence that long-term nutritional deprivation can promote longevity. Harrison et al. Discovered that treating mice with rapamycin, an inhibitor of the mTOR system, increased the median and maximum longevity of the mice a few years later. Selman et al. Also discovered that genetic deletion of S6 protein kinase 1, a component of the mTOR signaling system, extended the lifespan of mice and protected them from age-related diseases. To find pharmacological classes relevant to C. elegans aging, Yi et al. Created a pharmacological network. Resistance to oxidative stress and lifespan extension were shown to be grouped in a few pharmacological classes, the majority of which were connected to intercellular signaling. Putin et al. Also created a deep learning neural network that could predict human chronological age based on a simple blood test. Albumin, glucose, alkaline phosphatase, urea, and erythrocytes were found to be the top 5 most important blood markers for establishing chronological age in humans. Mamoshina et al. Also built a deep learning-based hematological aging clock with millions of patients using blood samples from Canadian, South Korean, and Eastern European populations. The findings revealed that population-specific aging clocks were more accurate than generic aging clocks in predicting chronological age and defining biological age. Based on the data from the drug age database, Berardo et al. Created a random forest model to predict if a chemical will increase the lifespan of C. elegans. Molecular descriptors and gene ontology terms were employed to construct the model. The random forest's feature importance measure was used to choose features. The best performing model was used to predict the class of compounds in the GDI-DB database with an AUC score of 0.80. This study expands on Berardo et al.'s work by looking at the usage of the drug age database to forecast molecules with anti-aging capabilities. The random forest technique was used to determine whether a substance will prolong the lifespan of C. elegans. Based on the data from the drug age database released by Berardo et al., 5 predictive models were built, each using distinct descriptor categories. Molecular fingerprints and/or molecular descriptors calculated from the structure of the substances in the drug age database were used to create the models. Variance and mutual information-based approaches were used to choose features. To our knowledge, this is the first time molecular fingerprints have been used to develop machine learning models based on drug age database entries. The best model was used to predict the class of chemicals in an external database, which included 1,738 small molecules retrieved from the drug bank database. Random forest is a supervised machine learning technique that consists of an ensemble of decision trees, each of which is trained separately using a random subset of data. Because it is resistant to overfitting on high-dimensional datasets with limited sample sizes, the random forest model is commonly used in chemo and bioinformatics activities. The chemical descriptors used in quantitative structure-activity relationship models can have a big impact on their quality and predictions. Descriptors are computer-interpretable digital or numerical representations of chemical information of molecules that are ideal for model creation. The molecular operating environment software was used to construct 2D and 3D molecular descriptors in this investigation. 2D descriptors are derived from a molecule's 2D structure and give data on its structural, topological, and physiochemical features. 3D descriptors, on the other hand, are derived from a chemical compound's 3D structure and include electronic parameters, quantum chemical descriptors, and surface-to-volume descriptors. Molecular fingerprints use binary vectors to indicate a molecule's structure, with 1 indicating the presence of a feature or structural group and 0 indicating its absence. The RDKit Python environment was used to generate extended connectivity fingerprints of 1024 and 2048 bits, as well as RD-Kit topological fingerprints of 2048 bits. Finally, the use of molecular descriptors in conjunction with ECFPs was investigated to choose a subset of significant variables for predicting the anti-aging capabilities of the compounds in the dataset. Researchers used variance and mutual information filter-based approaches. Only the training set, which contained 80% of the chemicals in the dataset, was used for this. The number of variables employed by each model was reduced as a result of feature selection, lowering the cost of computational calculations. For identifying the chemicals in the test set, the feature subset with the greatest AUC score in 10-fold cross-validation was chosen. When two feature subsets with the same AUC score were employed, the subset with the lowest standard deviation was chosen. With that, we have come to the end of the video. If you enjoyed the video, hit the like button and let us know your thoughts in the comments section below. See you again with another video. Until then, bye.